Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning. Today we will examine DNA evidence analysis and how genetic fingerprinting is used in criminal investigation. I will focus on the practical laboratory steps, interpretation challenges, and the statistical statements that make DNA useful in court. We begin at the scene. Proper evidence collection is the first step. Investigators must avoid contamination by wearing gloves and packaging items separately. A single touch can introduce foreign DNA. In one case study, a glove left in evidence produced a secondary profile that complicated the investigation. That illustrates how collection errors can undermine analysis. Back in the lab, we check chain of custody records before opening packages. Establishing a reliable chain of custody is essential to admissibility in court. If the record is broken, an otherwise valid DNA profile may be excluded. Extraction is next. We extract DNA from blood, saliva, hair roots, or touch evidence. The extraction method depends on sample type and condition. After extraction, we quantify the DNA to see how much template we have. Quantification guides downstream steps because low quantity can cause stochastic effects during amplification. Amplification is usually performed by polymerase chain reaction, or PCR. PCR targets short tandem repeat regions known as STRs. We amplify multiple loci to create a genetic profile. STR analysis is standard because it gives high discrimination between individuals. After PCR, we separate the amplified fragments by electrophoresis and detect alleles at each locus. The pattern of alleles across loci is the genetic fingerprint. The laboratory runs positive controls to show the reagents work and negative controls to detect contamination. Negative controls detect contamination in reagents or during processing. Interpretation can be straightforward for a single source profile. When the profile matches a suspect and quality metrics are met, we calculate a statistic. One approach is the random match probability, which estimates how often the profile would appear in a population. Another approach is the likelihood ratio, which compares two competing propositions about the source of the DNA. Complexity arises with mixtures, where DNA from two or more contributors is present. A telltale sign of a mixture is observing three or more alleles at a single locus. Mixtures require careful interpretation and sometimes probabilistic methods to estimate contributor proportions. For degraded samples like old bones or highly exposed material, nuclear DNA STRs may fail. In those situations, mitochondrial DNA can be useful because it is more abundant and resists degradation. Mitochondrial profiles are less discriminating between individuals, but useful for identification when STRs are not available. Another specialized tool is Y chromosome STR analysis. It targets male lineages and is useful in sexual assault cases when male DNA is mixed with a large excess of female DNA. YSTRs do not distinguish between male relatives who share the same paternal line, so interpretation must reflect that limitation. Probabilistic genotyping software has become common for complex mixtures. It uses statistical models to weigh possible contributor combinations and to compute likelihood ratios. Probabilistic genotyping reduces subjectivity and models uncertainty in complex mixtures. When we report results, we must explain assumptions. For example, the population database used for frequency estimates matters. Population substructure and allele frequencies affect random match probabilities. Courts now expect experts to justify their population choices and present results in understandable terms. Databases like CODIS allow investigators to search profiles for matches. CODIS helps link cases or identify suspects, but a database hit is investigative lead rather than proof. A suspect identified by a database still needs confirmatory testing and a rigorous statistical interpretation. Quality assurance is continuous. Accreditation, proficiency testing, and documented standard operating procedures reduce the risk of error. Casework laboratories also document software versions and calibration of instruments to preserve reproducibility. To summarize, DNA evidence analysis involves careful collection, extraction, quantitation, PCR amplification of STRs, separation and detection, and statistical interpretation. Special techniques like mitochondrial DNA and STR analysis help in difficult cases. Probabilistic genotyping and rigorous chain of custody practices strengthen the reliability of conclusions in court. That concludes the lecture. Please review the case study handout that traces a cold case solved with mitochondrial DNA and a recent sexual assault investigation that required probabilistic interpretation of a complex mixture.
Listen again to part of the lecture. Then answer the question.
Probabilistic genotyping reduces subjectivity and models uncertainty in complex mixtures.
This concludes the listening section. For more TOEFL practice tests and study materials, visit spinta.org.